Okay. We can see euchromatin and heterochromatins in the staining section of electron micrograph. And heterochromatins are dark, they are tightly packed, and euchromatins are light, they are loosely packed. So this also a little bit of another explanation. The light areas are very active, so they can provide codes to make proteins. Now, when it comes to a DNA molecule, and appearance of the nucleus, scientists trying to discover organization of DNA molecule inside the nucleus. As a result, a group of scientists led by Morgan make the word chromosomes. So it make the word chromosomes to explain the organization of DNA in the nucleus. They started to come up with the word chromosome. So we are going to learn the architecture of chromosomes. So how these DNA molecule packed into chromosomes. So let's start to learn what is this packaging and why it is important and what is the role of packaging inside this nucleus. First thing is, we're going to understand the complexity of the packaging. Now, complexity of the packaging, we can understand like this. Take a cell. So we take average cell. Let's assume average cell size is diameter is like a 15 to a 20 micrometer. And then the nucleus, it could be again 6 to 10 micrometer. This is the diameter of the nucleus. So we can calculate the volume of this nucleus when we know the radius. For example, radius is, let's say, 5 micrometer. Then we can calculate the volume. 5 r square, we can calculate volume. Then we're going to see what is the length of the DNA molecule.
Sir, your voice is little not clear, sir.
There's a little bit of Wi-Fi issues these days, I think. Now, can you hear?
Yet little bit not clear, sir.
Give me a second, I will check. Oh, that's fast. Right, what we're discussing, the size of the cell could be a 15 to 20 micrometer, and the nucleus could be 6 to 10 micrometer. Then we have to look what is the length of a DNA molecule. Now, length of a DNA molecule, human DNA length is total length that we take a DNA molecule and we take the next DNA molecule and we take the next DNA molecule and then we make a line by adding to one DNA molecule to another DNA molecule in one cell, in single cell. So it is 2 meter in one cell. Now you can understand the challenge. 2 meter long DNA molecule going to pack in 5 micrometer, 6 micrometer nucleus. This is something like this. This is a 40 kilometer a thread going to pack into tennis ball. Now, if you are asked to pack 40 kilometer long thread into tennis ball, you can understand the difficulty. So it's a very long 40 kilometer thread. You have to push into the tennis ball until all the length of the thread insert into the tennis ball. This much of a difficulty, this much of a task facing to pack DNA into the nucleus. So it's a very big task packaging the DNA molecule into the nucleus. How cell overcome this problem? Cell used a method to compress DNA molecule in very systemic way. It is not a random process. That means if we take a cloth, just push into the bag, ended up with few clothes. But if we take the clothes, nicely iron and press it, and try to find in an organized way, then we can pack many clothes in a bag. So DNA packaging is very careful process, and still scientists trying to understand some of the areas which we can't explain. So we should all understand the molecular biology. How old is molecular biology? The discovery of DNA somewhere around 1956. Now we are 70 years old. So 70 years old with this molecular biology, DNA like things. Still too early to understand many things. So largely missing areas are there. If you feel that you do not get it, if you feel that you uh, feel some kind of a lost in certain level, you can always ask, why is that? What is the reason? So then we can find the link if more links to understand these stories. So DNA packaging, the aim is to pack this 2 meter long DNA molecule into a 6 micrometer nucleus. So it uses a very systemic method to pack DNA in the nucleus. So let's start how this works. Now, after learning all these things, scientists proposed 4 levels. It packed in 4 levels. First, second, third, and fourth. Right? So we know well DNA molecule is like this. So this DNA molecule, we learned before, it's a double helix and long molecules consist of these 2 strands. So we can roughly, approximately, we can give this a diameter like a 2 nanometer range. Is the DNA molecule is 2 nanometer range. Okay, now we are coming to the first packaging level. The first packaging level, protein molecules serve as a scaffold to wrap DNA around it. So protein molecules are important wrapping DNA in the nucleus, because this packaging is not a permanent packaging. DNA should available for protein synthesis. So if it is very tightly packed and cannot recover again, that is also useless. It should be packed, but it should be also available when necessary to make the proteins, because DNA provide the codes to protein. So both sides should be handled, should be packed, should be also available for protein synthesis when is required. Right. Now the first thing is roll-up proteins. So what are the proteins? Protein molecules are called histone. The group of proteins are called histone. First, we will see how histone looks like. Right, so I try to draw a histone molecule now. Okay. One, two, three, four. We can see 4 units in different colors. Then again, I'm going to add another unit here, another unit here, and another unit here, and another unit here. How many units now? We have 8 units. 8 histone protein. Now in these 8 histone proteins, they are made out of different subunit, made with different subunit. I think if you recall the hemoglobin molecules, the hemoglobin molecules are made with 4 subunit, 2 alpha and 2 beta. So this protein, histone, is also a little bit similar. They have 8 subunit, and these subunits are named as H. So H is given for histone. H, A2, H, B2, H3, H4. So 2 of this, 2 of this, 2 of this, 2 of this. So they get together and join to make 1 unit, right? So they make 1 unit at the end. So I just draw it like this as a 1 unit now. Just to make it simple, I showed you as a 1 unit. Now this provide a space, a background or stage the DNA can wrap around it. Now our DNA molecule, it's just going to make a one turn around it and coming back from the behind, and then again going like this. So it is close to two turns, but it's not completely two turns, right? It is close to 1.7 turns. You can see two turns, but actual measurement is 1.7 turns. Okay, then there is another histone protein. This is another one. And now this DNA again going to the next one, and it is again going one turn and then going again another turn like this. So I can draw repeatedly like this to many times for the first structure, to show the first structure. Again another one maybe, and DNA is going again around it, close to 2 turn, 1.7 turn, like this. Now, when you look at this one, we can give a name, beads on string. So the first level is called beads on string. This is the first packaging, beads on a string. Now this diameter, close to 10 nanometer. So first level of packaging is beads on string, the 8 histone unit joined together, and this new unit has a name, nucleosome. Nucleosome is histone plus DNA wrap around it. So then nucleosomes serve as beads, right? So this is the nucleosome, and string is the DNA molecule. That is the first level of packaging. So then complete DNA molecule can wrap around this histone until it completely make this bead on string or necklace. That is the first level of packaging. Now here I add a good picture because it's very difficult to draw the 3D shapes. So you can see here 8 unit of histone, and then second histone molecule with 8 histone molecule and the DNA molecule wrap around it. So this represents the nucleosome. When the histone and DNA wrap around it, it forms the histone. And this part of the DNA molecule is known as a linker DNA. So it links one nucleosome with another nucleosome. That is known as a linker DNA. So this is the first level of DNA packaging. Beads on string, 10 nanometer long nucleosome molecules. Okay, now we are going to the second level. Second level is this again represent a 3D picture of the beads on string. A second level is known as chromatin fiber. Second level is known as chromatin fiber. Okay, let's see how chromatin fiber is formed, right? Now, histone protein, this is the histone protein, and the histone protein has 8 subunit and different names given: H2A, H2B, uh, H3, and H4. So if you look at this one, H2A, okay, H2 is there, H3 is there, H4 is there, H1 is missing. So when they give names, they give 2, 3, 4. But where is 1? Why didn't they start with H1? Yes, H1, then another type of histone protein. So now H1 is coming to this level.
Sir, can you explain that naming, sir, again?
So it's just easy: H1, H2, H3, H4. These are the name given to the different histone protein. And we can see H2 here, yes. We can see H3 here, yes. We can see H4. But H1 is missing. In the original nucleosome, we learn H1 is missing. Now H1 coming to the second level. So first level, we have H2, H3, and H4 histone protein. Now H1 is coming to the second organization, second level. So I'm going to draw now H1. So let's say this may be the H1. H1. Now DNA molecule again going like this. It should go 2 turns. 1 going behind it and going like this. So let's say this is H1 now. Now H1 is important to link the nucleosome. H1 histone is important to link nucleosome together. H1 histone is important to link nucleosome together. Now, if we go to this level, beads on string, they have a gap. The gap is, for example, this is a gap. This is a gap. Now, H1 role is to reduce this gap. They're going to bring all these nucleosome together. So then H1 play this role together with a linker DNA is the length from here. So linker DNA can still bend and assume that the nucleosome are coming together. So if it is come together, what we can draw here, so this is one nucleosome. So this is one nucleosome, this is one nucleo, this one like this. So they come together, and then we can also show the DNA is like this, wrap around it as usual, and then link like this. So then they're going to organize this nucleosome in a special way to make it very compressed now. Bead on a string is like necklace. Now imagine if you can find a necklace with bead, you can wrap this, spread it out, And then next turn, you're going to bring all the things into one place. You're going to squeeze into your hand. It's same thing, the nucleosomes are coming together to make the chromatin fiber. So H1 protein play a role to bring this together to make the chromatin fiber. So if we want to see how it looks like now, uh, it is like this. Don't worry about this different name, solenoid and zigzag, because scientists trying to make a model. After they bring together, this is the appearance and this is the appearance. So they give different appearance. So, but we can understand, like looking at this picture, all nucleosomes are joined together now. They're very close to each other. So this going to make this chromatin fiber. Then the chromatin fiber is the whole collection of these histone proteins like this. And this around 30 nanometer in the diameter. So if I draw this one, the DNA molecule, I can show something like this. Right, just to summarize again 1 and 2. The first one, histone used to wrap DNA around it to form the nucleosome. In the second level, these nucleosomes are joined together to make the chromatin fiber with 30 nanometer. So these are the chromatin fiber. A good picture, you can find it here, chromatin fiber when they join together. Now third one, third level. This fiber, chromatin fiber, make a loop, right? It's going to make a loop. Now I'm coming to that one. Third level is called loop domain. So this is the third organization, loop domain. Beads on string is number 1. Chromatin fiber is number 2. Loop domain is number 3. Right, let's see how this loop domain form. Now assume this is a chromatin fiber. I use the same color then. It can make a loop like this. And then again another loop like this. And maybe loop like this again. And loop like this again. So when it make the loop like this, still it needs some kind of proteins to provide the support, and these are non-histone protein. So they are different proteins. They are not belongs to histone group. They provide the scaffold, the skeleton, right? Scaffold protein. And this is the chromatin fiber. And this structure represent loop domain. So this provide the loop domain, right? So again, quickly we go through these 3 levels. Number 1, beads on string. Then the beads are joined together to form this chromatin fiber. Then chromatin fiber make loop to make the loop domain on non-histone protein. Now this loop domain move into the last and fourth level. last and fourth level. So last and fourth level, what is going to happen? This loop domain again going to a coiled. So this loop domain is going to make coil to make the fourth level. So here you can see again the same structure, 30 nanometer chromatin fiber from the loop domain, and the size of the loop domain, the diameter of the loop domain is 300 nanometer. So this
So what is the scaffold meaning, sir? That scaffold.
Scaffold is the consider as like a skeleton. Like you see the if you take the ball of or the the thread we use for the sewing machine, and this plastic one is the scaffold. This is the thread. For example, this white color or any color of plastic is the scaffold that is used to provide support. So this word scaffold indicate a supportive protein. So they are non-histone protein. So it provides the kind of a support that loop can make on it. And the size is 300, diameter is now 300 nanometer. So if you recall how this diameter changed, DNA is 2 nanometer and beads on string 10 nanometer, and chromatin fiber is 30 nanometer, and loop domain is 300 nanometer. Right? So 2, 10, 30, and 300.
So your network connection got stuck, sir. Now it's okay, sir.
2 nanometer, 10 nanometer, 30 nanometer, 300 nanometer. So here is again a picture taken to show it 30 nanometer chromatin fiber. This is a loop domain and non-histone protein chromosome scaffold. That means a supportive proteins used to make the loop domain. So we can understand by looking at this structure, we can further compress the chromatin fiber. If we stretch this chromatin fiber is long, but here we compressed. So loop domain help to compress the chromatin fiber. Now we are coming to the last level. Last level is metapase chromosome. Small remark, the resource book use mitotic chromosome. Correct word should be metapase chromosomes. Even the
So your network connection got stuck a bit, sir. So what did you explain in the two nanometer things?
is it did you check your one your network?
No, sir. Now it's okay, sir.
Because now I have a speed network. I see anyway. We'll check again. Other, yeah, it's quite high speed, 63 kilobytes per second.
Yes, now it's okay, sir.
Yeah.
Can it explain part again? Sir?
Okay, now the fourth level, the name given for the fourth level is mitotic chromosome in resource book. It should be better to write metapase chromosome because this word is used by textbooks. Metapase chromosomes. The fourth level is metaphase chromosome. And if we use a reference book as a Campbell biology, I think Campbell biology also use metaphase chromosome. So then fourth level is metaphase chromosome. Now I take this picture from Campbell Biology. It's a very nice picture that we can understand all three levels in one place. Now we learn the 30 nanometer fiber. So only missing is, I think in this picture, first level bead-on string. After that, second level is chromatin fiber. So chromatin fiber, you can see the whole histone proteins are joined together to make this chromatin fiber. And third level is the loop domain. You can see loop domain make this loop. And after that, this loop domain going to coil to make the chromosomes. So chromosomes is metaphase chromosome with 1400 nanometer. So what we learn so far, this loop domain, so it is going to further compress now to make the chromosomes. So this is a part of the chromosomes now. And in metaphase, usually we observe chromosome in mitosis clearly. And we can see chromosomes like this shape, X shape. And this one is called chromatid, and both together we call chromosome. So last structure is the metaphase chromosome is this one, and this is 1400 nanometer in diameter. So loop domain coiled to form metaphase chromosome. 1, 2, 3.